Okay, good afternoon, everybody. And first of all, thanks for having invited me to this meeting. It's very exciting in the city and the university and people going around to this university is very exciting and very young, as expected. So I'm a virologist. I'm involved in both the diagnostic and research activity at the National Institute for Infectious Diseases in Rome. And my first interest is on chronic viral infection, including HIV and hepatitis viruses. So first of all, I have to tell you, I fell in love with the next generation sequencing 3 years ago. And as a virologist, you may ask, why? Do you think that it's really important to have such a potent tool to investigate on microorganisms that have such small genome? What can be the usage of the application of next generation sequencing in studying such small microorganisms? And so this is the outline of my presentation. I will explain you something on HIV, going into the dynamics of viral quasi-species. I will explain what is quasi-species, taking into account the tropism of the virus, and then I will cover very, very shortly some aspects of HBV biology. So let's go to the quasi-species. So quasi-species is a phenomenon that now is very, very appealing, and is the name that was applied to virology to indicate the fact that several viruses, including mainly RNA viruses, but also some of DNA viruses, especially the HBV, that has a step in virus replication involving the action of a reverse transcriptase. So the replicative enzymes of these viruses are very, very error prone. So this means that in every replication cycle, a number of changes, mainly not fit for the viruses, are included in the virus genome, giving rise to not a unique genome circulating in humans or in the host, but a population of very, very similar genomes that are each different from each other for small mutations. And this is a particularly interesting field for the application of next generation sequencing. I will explain why now. Let's go into details of viral quasi-species. This is a graphic representation in a recent publication, where you see one virus is a founder virus. You can assume that this virus that is infecting the host. Each virus replication that is represented by the circles led to the incorporation of some mutation in the genome, so that according to the subsequent cycles of virus replication, you can see the progressive evolution of the virus in a complex mixture of viral genomic variants. Each of this mixture is very similar to the founder, but they are different for the incorporation of a small number of mutations. So along the subsequential virus replication, you can see that in the host, not only one genome is present, but a mixture of genomes that each differ from the other for a small number of mutations. And this population is what we refer to as quasi-species. And it is a property of RNA viruses. HIV is one of the most important viruses in this category, but also hepatitis B virus and other DNA viruses. Of course, this is an oversimplification of the situation, since the quasi-species may be very, very complex. And what is the advantage of having a quasi-species in one single host? You may imagine a quasi-species is something that is very plastic. It can adapt to different evolution pressure, so that if something like immune pressure or even pharmacologic therapy administration exert a evolution pressure, and the virus that has such a plasticity in virus genome can evolve and adapt and even occupy different replication space in one organism. What you can see at the end is that in one organism you can see different bodies, different times, where the quasi-species is slightly different. And you can analyze this, taking into account the potency of the deep sequencing. So, when using a sequencing approach, you can, let's assume that this is a quasi-species. You see viral particles slightly differing in color. This means that they are different, slightly different in the genome composition. If you make one population sequence, you only have what is representing the mean virus population. This is called the population sequencing. To approach the composition of quasi-species, classically, you have two different possibilities. One is to clone different genomes, and to sequence individual clones, so that you have, for each clone, one sequence. And you can say, this is one virus component of the quasi-species, the other clone is the other, another component of the virus quasi-species. You can also perform a limiting dilution PCR, and then sequencing the individual clones. This is a limiting dilution cloning, and the concept is exactly the same. What is the limit of this approach is that you only can sequence a limited number of clones. But if you have a very, very potent system tool, you can analyze a lot of clones at once, and this is what is the advantage of the next generation sequencing. So this is what you have with population sequencing, one consensus sequence. If you apply the clonal approach, you have 10, 20 sequences that you can have for each time point if individual sample. This is the graphical representation of a viral quasi-species. Each of these colors represent one patient. This is HIV. And the quasi-species analyzed is the GAG gene of the HIV. And you see in each patient, each color, you have different points, different components of the viral quasi-species at each time point. These are several patients analyzed at 2 time points after therapy interruption. By the way, this is not important. You can see that each patient is represented by viral quasi-species. This is in one patient, viral quasi-species in the ENV sequence. You see different colors represent different body compartments. You have a provirus in monocytes, in red, provirus in T lymphocytes, in green, virus, circulating virus with markers of monocyte replication in blue, and in pink, virus from T lymphocytes. And you see this is the graphical representation of the viral quasi-species. If you go to the next generation sequencing, what is the difference? You can enormously increase the number of clonal sequences you can analyze. You can go through 100 or 10,000 or even more clonal sequencing. I won't go into details of techniques you already heard about today. And what is also interesting is that you can also count the individual components, the individual sequences, how many times one viral quasi-species, one viral component is represented in the viral quasi-species. So you not only have a qualitative representation, but you also have a quantitative representation of the viral quasi-species. What is the result? If you see this is a quasi-species analyzed with the clonal approach, if you have the same sample analyzed with next generation sequencing, you have something like that. So what you see is that individual clones that are here in red, in blue, and so on, you can find them into the viral quasi-species, but what you get is an enormous increase in the resolution power of the viral quasi-species. And you can also highlight the presence of some of the components that are not highlighted by the clonal approach, since this quasi-species, these components of the viral quasi-species is very rare. It represents about 1% of the entire quasi-species. So to appreciate it with use of clonal approach, you should have at least thousands clones. Let's go to what can be done with this technique. First of all, I have to spend a few words on the HIV tropism. That is one of the aspects that I have deeply investigated with this system. So HIV is the cause of AIDS, of course you know, and mainly you can divide the tropism, let's say the cells that are targeted by the HIV, in two main components. One is the T cells, and the other main component is monocytes. What is different between these two cells is the presence of the co-receptors in addition to CD4, that is the main receptor. So the virus uses the second receptor to invade the cell and to replicate. So the virus that is able to infect the monocytes is called R5, that is using the chemokine receptor CCR5. So this virus is called R5. And the virus that using the alternative co-receptor, that is CXCR4, is called X4. There is a complex dynamics in the development of the evolution of the virus. What is usually and very simplistically assumed is that the virus, the R5 virus, is the virus that is mainly prevalent during the first stage of the infection, and it evolves progressively toward X4 using variants that are typically represented in the last stages of the disease. From the therapeutic point of view, it is important that a new class of antivirals have been developed called the R5 antagonists. That the prototype, and is already in the therapy, is the drug is Maraviroc, that is CCR5 antagonist, and it is able to block the infection of only R5 strains. That is, this drug is not able to block the infection by the X4 variants. So it's important to establish the tropism of the virus in one single individual to assess if the therapy with Maraviroc can work. And what is the genetic determinant of the usage of CXCR4 or CCR5 is the one portion of the GP120 molecule on the virus surface, and this portion is a small loop that is called the V3 loop, whose sequence is able to address the virus to the CCR5 or to CXCR4 co-receptor. So the sequence of this loop is able to give information about the co-receptor usage of HIV. Of course, there are several other main ways to establish the tropism. The main one of these is the phenotypic test that is using cells artificially engineered to express CCR5 or CXCR4 to establish whether the virus uses one or the other of the co-receptor. So these tests are called phenotypic tests. They are very costly. They are very insensitive, but they have been the basis of establishing the tropism for a while. But also the sequence can give information, and the so-called genotypic tests are able to give information based on the sequence of V3. And taking into account what I've said about the quasi-species, you can imagine that in one single individual, you can find viruses that have the sequence of V3 specific for the use of CCR5, or the viruses that are able to use the CXCR4. What is the approach we used to analyze, to go into detail in this analysis using the 454? We used the amplicon approach. Let's say we identified the V3 loop, designed the system to amplify, PCR to amplify this region in forward and reverse, then analyzed the PCR product by the 454. And this is what is typically a flow-gram. And it is important to say that when analyzing virus quasi-species, it's important to avoid the errors, sequence errors inherent of the system. That is, you know, pyrosequencing is also prone, is very prone to error, mainly in the region where there is a homopolymeric stretch. We used an algorithm that was originally ideated in our laboratory that say this is what we obtained at the beginning, then we made the translation in amino acid. The translation was used to eliminate all the sequences that carried the stop mutations. And we assumed that these mutations were artificially introduced by the system. So in this way, we eliminated all the reads that were not coding, so leading to truncated products. And usually we throw about half of the sequence of the reads we obtain. Then we trimmed the sequence to obtain just the piece of amino acid that was coding for V3, and even we added another step for controlling the accuracy of the sequencing by using only the reads that had at least one forward and one reverse read. In this way, we were able to reduce the error rate, and so we were able to establish a threshold of sensitivity of variants that can be set to about 5 times the error rate of the system. Let's say this is 0.6%, and we establish 0.3% as the sensitivity of our system. What we do with the sequences? We plot these sequences, again, algorithms that are able to to predict the co-receptor use of each of these variants according to an algorithm. The algorithm we use is a PSSM that is available freely on internet, and based on this system, each of the sequence obtained by the 454 was transformed in one score, and the score was proportional to the usage of co-receptor. In this way, I give you a graphic representation of the quasi-species. So here it is the score of the viral quasi-species, and in the in the vertical axis we have the frequency of the individual variants, and you see just to see that prototype R5 and prototype X4 using variants have a score of about -13, for instance, R5, and +3. For instance, for X4. That is here for R5 using, and here for X4 using. So you can see that, for instance, the virus circulating as RNA in a patient, in this case, has a is composed mainly of R5 viruses. In this window, this is the window of uncertainty. That is, all the variants that are on the left are R5, and all the variants on the right are X4. This means if we made the same analysis, not on the virus circulating as RNA, but on the virus archived as DNA in the lymphocytes, we can see sometimes that there is a very different situation as compared to virus circulating. For instance, in this patient, we can see that while in the circulation only 0.3% of the virus is X4 using, in the virus archived at the proviral level, we have a quasi-species that is different from that are in the circulating virus. That is, in the proviral DNA, there is a consistent, high proportion, 27% of the sequences that use the X4. What does it mean? I will skip this. If we compare the quasi-species at RNA and DNA level, we can see typically that the quasi-species archived at DNA level has a higher degree of heterogeneity as complexity and diversity of viral quasi-species. And this is what is expected, since during the evolution of the HIV within an individual, you see that the virus is continuously archived at proviral level, and so the provirus represents a much diverse quasi-species, taking into account all the viruses that are producing during the various stages of disease. While when you analyze the virus RNA, it is the replicating virus. This means that you can have one of the quasi-species that is at viral DNA level that is replicating at that moment. So, from this preliminary analysis, we can say that the ultra-deep biosequencing combined with the genotypic algorithm for co-receptor usage prediction provides a detailed quantitative description of HIV quasi-species in the V3, that is a quantitative description of the quasi-species for the co-receptor usage. Usually at the DNA level, the quasi-species is more complex than that is replicating in each given moment along the disease history. And what was unexpected is that at the level of proviral DNA, we see often a bimodal distribution of viral quasi-species, that is X4 virus and R5 viruses that are archived, but often in the circulation we only see R5 virus. And recently there is a great enthusiasm about this argument, since the prediction of co-receptor usage is something that is important for therapy. I have said already that the administration of Maraviroc is only possible when the patient has R5 using viruses. But this assumption is based mainly on circulating virus. What happens if you take into account the virus archived in viral DNA? That is something that up to now has been just disregarded. So, in addition, what is I can summarize a number of unexpected things that we could observe using this potent system when applying it to the viral quasi-species. First of all, usually we see more diverse viral quasi-species in monocytes as compared to lymphocytes. I will show you the details. But the virus archived in monocytes is replication competent. Some minority variants that are X4 are replication competent. If you take patients that are suppressed for a long time with antivirals, you see that the viral quasi-species archived in DNA in lymphocytes and monocytes can be very divergent. If you take out the therapy, you see that the virus starts again to replicate, and there is a mixing of the viral quasi-species between the two main reservoirs. And also seeing in acute infection, we have an important observation, and there is a correlation between the presence of high diversity and the need of therapy, that is, there is a more advanced disease. Let's say more into details. Okay. This is a patient, viral quasi-species, ENV quasi-species, in a patient who has been under suppressive art for a long time. So there is no massive replication in the circulation. AC in green is the viral quasi-species in lymphocytes, and in red the viral quasi-species in monocytes. You see that the viral quasi-species is quite separate in lymphocytes as compared to monocytes. And in addition, in monocytes, while the majority of the virus has a score for PSSM negative, that means that is mainly R5 virus, you can see small branches of the phylogenetic tree. There are quite divergent clusters of viral sequences that are using the X4. In fact, the score is positive. These minority variants archived in the lymphocyte, in the monocytes in patients with a long time suppressive therapy is replication competent. In fact, if you analyze the viral quasi-species in proviral DNA after interruption of therapy, you can see, for instance, this small cluster that is the same I underlined before in a tree that it's in a different graphical presentation. You see that this small branch that is the virus that is X4 while the majority is R5, after interruption of therapy passes to lymphocytes, that it is the blue circle. This means that the virus archived as a minority quasi-species in monocytes is replication competent and can spread. So this means that we have to take into account the virus archived for predicting the co-receptor usage and for deciding whether to use or not maraviroc as a CCR4 antagonist. If you leave the patients without therapy for a long time, you see that progressively the X4 variants are lost. This has a pathogenetic implication. In fact, one was thinking at the beginning, the majority of researchers were thinking that our X4 virus is more fit than R5 virus. This is not true. In fact, when you look at the patient progressively analyzed at different time points after therapy interruption, you see that there is some X4, this is percentage in vertical, the percentage of X4 using viruses, that is at the beginning you see a proliferation of X4 variants, but they are progressively lost. This means, or at least they are diluted by the overgrowing R5 virus. This means that the X4 variants are not favored into the organism.
5 minutes.
Okay. And we also can see that the virus, the X4 using variants is frequently found during acute infection. This is unexpected. In fact, we were thinking that during acute infection only the R5 virus was transmitted. This is not the case. We compared 20 patients, 10 of them, group A, needed antiviral therapy early in infection, and 10 did not need. This was based on clinical decision and mainly on the number of CD4 in the patient. You see very low here and very high here. This is the patient group that needs therapy. When analyzing by ultra-deep biosequencing the virus in these patients, we could observe that the patient group that need to be treated has a more diverse virus. In fact, the diversity is higher, about 10 times higher than the diversity in the patients who did not need therapy, and the percentage, the frequency of X4 variants was much higher in the patient who needed therapy as compared to patients who did not need therapy. You can see high, in some of these patients who needed therapy, even more than half of the circulating virus that was X4 using. When looking at the phylogenetic trees of these patients, you see that the patients who needed therapy that have a more diverse viral quasi-species, you can see graphically represented the viral quasi-species. You see the tree are very complicated. And on the contrary, when looking at the patients who did not need therapy, you see that the quasi-species is monophyletic. That is consistent with the very, very low variability of this quasi-species and with the very low frequency of X4 variants. And lastly, there is a great interest on HIV quasi-species based on the analysis of ultra-deep biosequencing. I would just go through the hepatitis B. I won't go into details since there is no time. Just I will go to the sorry for being so quick. I will skip this and I go to the conclusions just to say that the analysis of HIV quasi-species unveiled a very, very unexpected pattern for in viral dynamics of the quasi-species concerning, for instance, the I.N.M.I sequence, so the tropism. HBV was mainly investigated for the presence of mutations able to provide resistance to antivirals. And this is a very promising field also in HIV. And in conclusion, recently there are some interesting papers, review papers, dealing with the clinical utility of applying next generation sequencing to the study of these chronic viral infections. And there are a number of points that need to be further investigated. In fact, this is applied to HIV, but can be easily applied also to other viruses, including HBV. So the biological and clinical question facing the field of genotyping with ultra-deep pyrosequencing is the need to define the level of sensitivity needed, required to have a clinical utility, the effect of different resistant variants that have on treatment response, and requirement of having put together mutation in the same genome. As I told you, the genome piece that we analyzed is very short, and this is a limitation of the next generation sequencing, that at most you can have pieces of 400 base pairs. But for instance, the genome, the regions involved in antiviral resistance is more than thousands. So you have to apply very complicated algorithms to build to obtain contigs, and this argument is called haplotype reconstruction. That is a chapter of bioinformatics that is very complicated. And under very conclusion, we can say that minority variants carrying resistant mutations or even very biological properties arise spontaneously. They are present in the virus without any treatment, without any selection pressure. But the plasticity of the virus genomes is able to enrich or wash out this sequencing according to the external situation. And we must study very much to understand which is the clinical significance and which are the threshold to be used for deciding individually which therapy or which prediction of the virus evolution derived from this situation. And at the end, I want to acknowledge the very big group of people who made these studies possible, and that is people at the Laboratory of Virology in my institute that I'm leading, people working with HBV and with HIV. These are the bioinformatics, the clinical divisions of our physicians that provided the patients for the studies, collaboration with the group involved in virus resistance, led by Carlo Federico Perno, and finally the group of the bioinformaticians at the Inter-University Consortium that is called CARPUSPUR, that made possible to make a lot of advancement in the bioinformatic elaboration of the data that for the first time have been applied to virology in our collaboration in Rome. Thank you.
Thank you for a nice presentation of the application of this sequencing in viral genomics. And there is time for a fast question. Only one.
Why do you assume that the mutations leading to transcriptome are not real and they're actually generated by genes?
So this is an assumption. We made this decision since we wanted to define the co-receptor usage, and this means that you must have a functional ENV. For this reason, we decided to use only coding sequence, only sequences giving product, amino acid product, to be interacting with the co-receptor. We assumed that the genomes that are not giving functional products are either errors or unfunctional. So to analyze the quasi-species This is just for a functional point of view. We decided to
I
throw
understand,
out.
but those genomes could actually be co-packaged with other genomes and be carried and be very real, and maybe the basis for other mutations that give rise to other things. So
Okay,
I'm not sure I would discard the things just because it's known by design. I mean, because we think it's nonsense. That actually could be wrong.
Okay, we thought a lot, we discussed a lot about the algorithm to be used for correction, and there are a number of different orientation. For instance, in the study of resistance, the developed algorithm are not based on this assumption. But let's say this was an assumption, so it's open to reanalysis. But anyway, we wanted to avoid really the sequences that were not giving rise to a functional product, although they can be harbored by the viral quasi-species.
Thank you. Thank you, Maria Rosaria Capobianchi.